Okay, today we're going to go through energy in food, photosynthesis set 2. This is an extension of your learning for photosynthesis. Alright, so we are going to look at your open-ended questions first. Alright, first one. Now, whenever you do open-ended questions, you need to do annotations because the annotations will help you to make better sense of what is happening and what topic it is. So because this is a topical practice, it says photosynthesis and energy. So you know all the questions is going to be related to that. But in a full paper, you will not, right? A full paper has many topics. So you need to first identify what are the keywords or ideas in the question that is related to a particular topic. Identify that topic. Then from that topic, identify what concepts are being tested. So when you do that, you actually narrow down what kind of concepts you need to actually write. What will be the demands of the question? So Judy wanted to find out if the presence of light will affect the amount of gases taken in and given out by plants. So straight off the top, I have words like light, I have amount of gases, and I have given out by plants. So straight away in my mind, the word photosynthesis should occur because I know that during photosynthesis, there is an exchange of gases whereby carbon dioxide is taken in and oxygen is given out. Okay? So she prepared two set up P and Q as shown in the diagram below. She gave the plants the same amount of water and placed both set up in an open field from 7 a.m. to 10 a.m. Now the open field here signifies that there is actually sunlight. Okay, it is not there for fun. It's there to tell you that, oh, there is sunlight. And then after that, remember this. In every experiment, there should be one change variable. You need to identify the change variable. Okay, so the change variable in this case would be your glass jar. Now, so what does this then translate to? It translates to that one has light and one does not have light. Alright, and that is the difference. So based on what you already knew about photosynthesis, you should know that, set up Q, there is no photosynthesis. Whereas for set up P, there should be photosynthesis. Okay, now the graph below shows the change in the amount of gases in setup P throughout the experiment. Label the gases in the boxes provided. Okay, so take a look at it. Now, what does 7 a.m., 1 p.m., 7 p.m., 1 p.m. means? Think about it. What could change during the day that could affect photosynthesis? So we know at the top of our mind, I always tell my student this, okay, multiple times. Whenever you deal with photosynthesis, in fact, in your exam, because there will be photosynthesis questions and respiration question, there are two-word equation that you should write straight off the top, okay? And the one of it is actually your photosynthesis word equation, whereby you know that carbon dioxide together with water in the presence of light and your chlorophyll will give you oxygen and glucose or sugar. But the more correct name for it, the scientific name of it is actually glucose. But definitely sugar, no issues. So ask yourself, which of this variable that could affect the plant rate of photosynthesis change throughout the day? And your answer would be actually light. Right? Because at 7 a.m., your light is actually the lowest. And then around 1 p.m., your light should be the highest. And then after that, at 1 a.m., it should be the lowest again. Right? It's in the middle of the night because it's the open field. Sun is gone. So another one more thing that you need to take note of is this is actually an enclosed jar. So because it's an enclosed jar, right, the air composition inside does not changes in accordance with the surrounding. Okay, so whatever air is trapped inside is trapped inside. So therefore, it will reflect this change. Now, think about it. when it's photosynthesizing, your plant will take up carbon dioxide, correct? So, if you look at it, your carbon dioxide should decrease the most at 1 p.m. Why? Because that's where rate of photosynthesis is the highest. Because your sun will be the brightest. So because of that, your carbon dioxide would drop. Because your stomata would open the biggest to take in most amount of carbon dioxide for maximum amount of photosynthesis. And what happens is that the opposite is true for your oxygen. Why? Because when you photosynthesize, oxygen will be produced. So at 1 p.m., when you have the most amount of light, you also have max photosynthesis. Right? When there's max photosynthesis, you would actually get most amount of oxygen. So that's how you use your word equation to help you and remind you. Next, give a reason why at the start of the experiment, okay, the plants were placed in jars of the same size to ensure a fair test. Now, I've told you this many times, umpteen times. There are 3 things that you need to know. The 3 things that you need to know would be fair test, okay, reliability, and the last one would be actually accuracy. Now, these 3 things refer to different parts of the experiment. For fair test, it talks about your change variable being only one change variable. For reliability, it talks about is your results, can your results be repeated? And the last one, accuracy, talks about your measured variable. Okay, so what are the 3? I'm going to roughly talk about it. So in fair test, in order for a test to be fair, there can only be change or one change variable. Because when you have 2 change variables, you don't know your results, whether is it due to A or B. Let me give you a very simple example. So you want to actually find out if sleeping more will make you taller, correct? So in order to conduct this experiment, everything else needs to be the same, including the person. So technically, you will need to have a twin whose DNA is identical. They go through everything the same. They eat the same food, they go through the same, everything is the same. The only difference is the number of hours that they sleep. So if one twin grows taller than the other, you can say that, because the only change variable was sleep, the sleep must be the factor that affected height growth of height. However, if let's just say you not only change the amount of sleep, you also put in another factor. For example, food, the type of food that they eat. So when one twin grows taller than the other, you don't know if the additional height growth was due to the amount of sleep or the type of food. That's why it's not a fair test. Okay? I hope you get it. Reliability means how many times can you get back the same results. Okay? Because if you cannot produce or reproduce the same results every time you conduct the experiment exactly the same way, your test is not reliable. Just think about it in this way. COVID vaccination. It needs to be very reliable because they have to prove that it actually works. So it doesn't matter how many individuals you inject the COVID vaccine in, they should actually get a very high rate of efficacy. Means that it works many times. So maybe out of 1,000 times, okay, 999 times, okay, the vaccine will work against the virus. That is called a high reliability. okay, the results can be replicated. But that doesn't mean that the test is fair, neither does it mean that the test is actually accurate. So if you do something wrongly, okay, but you keep doing the same mistake, you probably would get a very high reliability. Because you did everything the same, so you make the same mistake, you're probably going to get the same results. But that doesn't mean that the result is fair. Okay? Yes, doesn't mean that it's fair. It's reliable, but it's not fair. Next, accuracy. So accuracy, right, we are talking about measurement. Okay, measured variable. So for example, let's just say I want to measure, okay, one end of the classroom to the other end of the classroom, right? I'm actually measuring the length. So there are different things that I can use. I can use a ruler, okay, your normal ruler, which whereby the maximum is 15 cm, or I can choose to use a measuring tape. So which one is more accurate? The measuring tape. Because every time you try to use the short ruler and to actually reuse it again, you may move a bit. It's not very accurate. Similarly, if I'm going to actually clock an athlete during Olympics games, I wouldn't use an analog stopwatch. That means the manual kind, because decimal places is only 1. So I can only see the decimal places up to maybe say 12.1 second. But if I use a digital one, it is accurate up to 2 decimal places. So it could be 12.15 second. Right? So using a digital watch would be more accurate. Okay? So this is something that you need to understand. Okay? Now, so give a reason why at the start of the experiment, plants were placed in the jars of the same size. So it's not, you cannot just say it's to make sure the test is fair. You have to tell me which aspect of it. So the same size of the jar, what does it ensure? It talks about the volume of gas, right? It doesn't matter to me, My main point, as in the size of the jar doesn't matter to me if I have another way to ensure that the amount of gases that both plants receive is the same. So this volume of gas being the same, the idea of it needs to be in my answer. Okay? Alright, so you need to say that this is to ensure that both jars have the same amount of oxygen and carbon dioxide. Alright, at the start. Because that will affect your results. Correct? You think about it. Because if you have a jar with more carbon dioxide, it could mean that the plant has an unfair advantage simply because there was a higher amount of carbon dioxide. Okay? Next. It was observed that for setup Q, the amount of oxygen decreased while the amount of carbon dioxide increased at the end of the experiment. Okay? Explain why. Alright? So, very simple, 2 things. Okay? Now, they told you, set up Q. So you look at set up Q, right? It's got no light. So if no light means what? Totally no photosynthesis. Okay? So they tell you towards the end, right? What happens is that carbon dioxide actually increase. They ask you why at the end of the experiment. So carbon dioxide increase tells you that there must be some photosynthesis some respiration that's taking place. Because when you respire, you give out carbon dioxide. Alright? Now, here is the key thing. You need to name the processes that cause this change. The first part is actually, okay, respiration. The second part is actually no photosynthesis. Okay, so the first thing would be there was no light. And so photosynthesis cannot take place. Alright, so when it cannot take place, what does the plant do? The plant can only respire. Now, you need to be very careful. The word still needs to be there because the plant is always respiring. Okay? The plant continues or is always respiring. So it took in oxygen and gave up carbon dioxide. So this part about giving up carbon dioxide links it to the increase. This part about oxygen tells you about respiring, the respiration. But do you see the processes? Okay, photosynthesis and respiration needs to be named. Okay, now because they ask you why was the results opposite? Do you realize they're asking you why is it opposite? Photosynthesis needs to be there. Because in P, the plant actually managed to photosynthesize. In Q, it didn't. That's why, even though it was just respiration that causes the change, you needed to include photosynthesis also. Okay? Because it's a comparison question. Alright? Next. Jane prepared the following setups as shown using similar plants. Okay? So once again, one is a clear box and one is a black box. So what does it translate to? It just simply translate to light and no light, my apologies. So no light and light. So this one, no photosynthesis, yes photosynthesis. That is how you must interpret it. And of course, the term photosynthesis should be in your answer. So she placed the setup near the window. The table shows the volume of water and the setup over the 5 days. Now, sometimes even before looking at your answers, I like to tell students, you should anticipate what the trend is. Okay, ask yourself, so which one do you think would lose more water? And my answer to you would actually be B. Because B actually is able to photosynthesize. And if you go back to your word equation, you know that water is being used up during photosynthesis. So this would tell you that your B should actually have a lower amount of water. Let's see if I'm correct. So true enough, you compare, it's lower what? 60, 100, 80, 130, 150, 100. So whatever the case is, okay, they all started off the same. I ended up with B being lesser than A. So was my assumption correct? Now, why did I ask you to do that? This is to frame your mind so that you know what you want to write in the next upcoming question already. Identify that process is occurring in setup B but not in the plant in setup A. Oh, photosynthesis, right? It's very straightforward, right? I don't even have to think because I think already. Based on the information above, okay, explain the difference in the final volume of water for setup A and setup B. So you have to state what is the difference in the sense that which is lower, which is higher, and then you also need to tell me what is the process that caused it to be different. So looking at this, the change variable was actually the lack of light. So you must tell me that. So I would say that unlike setup A, the plant in setup B was able to receive light, and hence it is able to photosynthesize, unlike the plant in setup A. Alright? Then after that, as water is required for photosynthesis, the roots of the plant in B would have absorbed more water, okay, for photosynthesis, resulting in a lesser volume of water left in the jar for B. So I'm very clear like that, step by step. Okay, it is not good enough to just say, oh, because B photosynthesizes, okay, and A doesn't. So therefore, B will use up more water. No, you have to tell me the roots are there for a reason, not for fun. You realize that? So you have to say the roots will have taken up, okay, more water. See, so many lines. Okay, so I would say that plant in B is able to get light for photosynthesis. Since water is required, okay, wait, sorry, I have to put unlike A, so we know. Since water is required for photosynthesis, the plant in B takes in more water resulting in less water left in its jar, okay, which is jar B. So it's very clear cut. Okay, you explain what is the difference between the two setup, the change variable, what does the change variable lead to, which is one is able to photosynthesize, the other cannot. And then you also explain that because water is required for photosynthesis, therefore more water is taken in by the roots. Okay? Sorry, by the roots. Let me just add in here. This makes it very clear. Next question, part C. Using setup B, Jane applied oil on both sides of the leaves. Now, a common misconception that students have is that they think that oil is opaque, unless they tell you it's crude oil. Crude oil is the kind of oil that you see in the seas that when there's oil spill, it's black in color. So for normal type of oil, it's actually transparent. Okay, it's colored but it's transparent. So it would still allow light to pass through. Now what it does not do is that it does not allow your gaseous substances to go through. Things like your oxygen, carbon dioxide, all cannot pass through. Okay? They tell you that it's applied on both sides of the leaves, stem and the leaf stalk on the plant, and left the setup near the window. So near the window tells you that there is still light for photosynthesis, but that doesn't mean that it's able to photosynthesize. Because if you remember correctly, based on our word equation over here, you know that you would actually need carbon dioxide, water, light, and your chlorophyll in order for photosynthesis to successfully happen. So in here, you may have your light, but you may not have the rest of the items that you need. So the plant in setup B could not carry out photosynthesis and the leaves turned yellow. Okay? In fact, they were very nice to tell you that they could not carry out photosynthesis. You need to assume that they may not. okay, because of the coating of the leaf. I'll explain why in a little while. Okay, so they say explain why plant B could not carry out photosynthesis. Now the reason is this, because it does not allow your oil, does not allow carbon dioxide to enter, okay, you will not be able to photosynthesize. Now it is also very important to remember that your stomata has two functions. The first function is to lose water in the form of water vapor, because only when water vapor is lost, it can pull the next water particle, and then the next particle will pull. So it forms a continuous chain, allowing water to be drawn from your roots all the way up, okay, to your plant. Now, the second function would be gaseous exchange. Now, when you actually state that the function of stomata is for gaseous exchange, it is not good enough. You have to tell me for what. So you have to say the stomata takes in carbon dioxide for photosynthesis and gives out, okay, takes in carbon dioxide for photosynthesis, and it also takes in oxygen for respiration when the plant is not photosynthesizing. Okay? Alright? So you need to first mention that the stomata of the leaves are blocked by the oil. Okay, the oil. Now, because in this case, there's only one plant, the question was being very specific. So you don't have to say, oh, the leaves of plant B. Okay, there is only one plant, right? Therefore, carbon dioxide could not be taken in for photosynthesis. Alright? Next, explain why a layer of oil was poured on top of the water in setup A and B. So basically, this is to prevent the loss of water in the speaker, right? Just double check. Yeah, in the container. Okay, in the container. Via evaporation. Okay, now there is an alternative way you could answer this. You could say is that if this is to ensure that water is only lost via the roots of the plant. Okay? Next question. This is another experimental question. So you realize that a lot of questions are experimental. The first thing that I always do, like I tell you, is not only just to identify the topic and the concept, but to identify the change variable as well as the measured variable. Okay? So Gwen conducted an experiment in a dark room using two setups as shown. In a 24-hour period, the lights were first switched on for the first 8 hours and then switched off for another 8 hours, and then finally switched on for the final 8. Okay, so that means if you think about it and you see things like plant and then you see light, so basically if there's no light, there confirmed cannot be any photosynthesis. But if there's light, there may be no photosynthesis also, depending. Okay, because photosynthesis does not just depend on light only. So you need to be careful enough to make sure all your other factors for photosynthesis to occur are there. The liquid indicator turns yellow when there is more dissolved carbon dioxide in the water, and turns purple when there's less dissolved dissolved carbon dioxide. Now, when it's very wordy like that, okay, a tip that I like to do even for myself is to write it out somewhere so it helps me. So I know that when there is more CO2, it becomes yellow. And when there is less CO2, it becomes purple. Right? If not, I would have to keep reading that sentence again, which is not only time consuming, but it could be very confusing. Alright? So the table below shows her results. So even before you look at the results, I always tell you, it'll be good to actually think about how should the result be like. Okay? So one thing, what are the change variable? The change variable would be the plant, right? This is the CV. And what is the MV? The MV is actually the amount of CO2 that you're measuring based on the color, the color indicator. So just based on this, straight away I know that the plant, when light is on, should basically reduce the amount of carbon dioxide, and hence it should be purple in color when the light is on. And when the light is off, there should be an increase in carbon dioxide. Now, here's the thing. If I look at it, the fish, it should always be yellow, because there is no plant to actually take in the carbon dioxide to reduce the concentration. But it will always be yellow. Now, students tend to think that because there is no, your plant, right, it doesn't respire. No, your plant also respires. So if I were to do a comparison, if there's something as yellower, your setup A will be yellower when the light is off, because you've got two organisms here. So it depends whether or not your color indicator is a quantitative one. Quantitative one means besides telling you increase or decrease, it can tell you which one increase more. Okay? So in this case, it doesn't, because it's either purple or yellow. So based on it, I know that when my lights are on, this should be purple, decrease. Why? Because I know that the plant takes in the carbon dioxide for photosynthesis. I'm correct. And then B is always yellow all the way, like I tell you, because the plant, the fish is forever respiring, is forever taking in taking in oxygen and giving out CO2, so it will go up. Okay? And then when the lights are off, it's yellow for A, and then when its light is on again, it will start to actually decrease again, because your plant will start to take in your CO2. So this is consistent as to my initial assumption. Now, I will know how to answer. Because if I can predict correctly the results, it must mean that my hypothesis is correct. And I can use that to answer. Explain why the color of the indicator in setup A changed from purple to yellow and back to purple. So you will need to say that, unlike, I know they never ask you to compare, but it's good to say, unlike setup B, setup A has a plant that can absorb. Let me make this a little bit more substantial. Okay, so unlike setup B, setup A has a plant. So when the light are switched on, the plant would take in dissolved carbon dioxide hence decreasing the amount of CO2 in the water. Okay? When the lights were switched off, both, this is where I'm going to bold this in a different color because students tend to miss out this part. Both plant and fish respire, giving out carbon dioxide. Now, in this case, my answer didn't talk about oxygen at all. You realize that? Now, the reason is because the indicator, the color indicator, only finds out for, or is only concerned about your carbon dioxide. So, since it doesn't test for oxygen, there's really no need for you to mention oxygen if you realize that. Okay? At the end of the 24-hour period, Gwen observed that the fish in setup B was staying near the surface of the water, while the fish in setup A did not. Explain her observation. So think about it. What is the difference between these two? One has a plant and one doesn't. So you think about it, when you actually, the plant is able to photosynthesize. So when you photosynthesize, what is it that your plant gives out? It would be oxygen, right? So that will tell you that this tank over here has more dissolved oxygen. And be careful, the word here is dissolved oxygen. So over here, because there is no dissolved oxygen, your fish is going near the surface to get atmospheric oxygen. Alright? Now, be very careful. Many of the students, they would term it as setup B has no oxygen. And that's why the fish will go to the surface to get oxygen. Now, this cannot be factual. No means absolute zero. So what we are looking for is insufficient. Insufficient is the word, or not enough. Okay? So they ask you to explain. So you would say that, so you're talking about, you want to talk about, okay? Why is it? So you must talk about both. So you can choose to talk about A or talk about B first. So I will actually follow the sequence. They talk about B, so I will talk about B. Okay? So I would say that in setup B, there is no plant to carry out photosynthesis. Hence, there is insufficient dissolved oxygen for the fish to respire. Hence, therefore, the fish swims to the surface to get more oxygen. Okay, so this part is important. You cannot just say not enough oxygen and stop there. No. Why then it goes to surface to get more oxygen? Then the next part you have to talk about A. In setup A, there is a plant to photosynthesize and produce oxygen for the fish to respire. So it's very clear. You make a comparison, right? Because that's the requirement of the question.